Assalamu alaikum dear students, welcome to Dr. Roomi lectures. In today's session, we are going to study journal features and functions of red blood cells. Red blood cells, also called as erythrocytes, are most abundant of all the blood cells, being more than 99% of all the blood cells. Most important function of red blood cells is to transport oxygen. But it's very interesting to note that oxygen gas is poorly soluble in our blood plasma. Just 3% of the total oxygen which is carried by blood is dissolved in plasma. So where is the rest of 97% of oxygen in our blood? That 97% oxygen is bound with hemoglobin. Hemoglobin is a pigmented conjugate protein which is present inside red blood cells, and this increases the oxygen carrying capacity of blood. 97% of the total oxygen carried by our blood is bound with hemoglobin. In some lower animals, hemoglobin protein is present free in their plasma. However, in human beings, this hemoglobin protein is present with the red blood cells. You may be asked this question in viva voce examination. What is the advantage of having hemoglobin packed within red blood cells? The advantage is that in this way, hemoglobin will not be filtered out of glomerular capillaries. You see, this hemoglobin molecule is a small molecule relatively, and the glomerular capillaries are very permeable. Every time when blood will pass through kidneys, in each cycle, 3% of the hemoglobin will be filtered through glomerular capillaries. In this way, hemoglobin will go out of body. So, just to keep hemoglobin conserved within our blood vessels, we need it to be packed within red blood cells. Let's talk about the shape of red blood cells, which is very well suited to carry oxygen. Red blood cells are like flat biconcave discs which are indented from both sides. When you see this red blood cell from a side, it looks like a dumbbell. So red blood cell is like a donut which has a flat center instead of having a hole in it. Its diameter is about 8 micrometers, and the thickness is 1 micrometer in the center where it is thinnest, and about 2 to 2.5 micrometers at the edges where it is thickest. As compared to the flat biconcave shape, the shape of red blood cells becomes spherical in a disease which is called as hereditary spherocytosis. As compared to the spherical cells which we see in hereditary spherocytosis, these flat biconcave cells, they are better in diffusion of oxygen and carbon dioxide. It's because the hemoglobin which is packed in these cells will be very close to the surface. You can see here in the center and also at the edges, because these cells are not spherical. In case of sphere, the hemoglobin which will be in the center will be too far away from the surface, so it will be difficult for the oxygen and carbon dioxide to diffuse. But in this case, the oxygen and carbon dioxide, they can easily diffuse. The mean volume of one red blood cell is called as mean corpuscular volume, and its range is between 80 to 100 femtoliter. On average, its value is 90 femtoliters. When a red blood cell is smaller in size, then its mean corpuscular volume is less than 80, and we call it microcyte. When its volume is more than 100 femtoliter, then we call it macrocyte. In iron deficiency anemia, the red blood cells are smaller in size, so they are microcytes. In folic acid and B12 deficiency anemia, the red blood cells are bigger in size, so that's why they're called as macrocytes. These biconcave discs of the red blood cells have a lot of excess cell membrane, and when they are passing through narrow tortuous capillaries which may have a diameter of as less as 3 micrometers, they can deform to any shape. They can squeeze remarkably, and they do not rupture. In order to carry more and more oxygen, we need to pack more and more hemoglobin inside this bag of red blood cells. And this is done by not having nucleus and other organelles in the red blood cells. When these red blood cells are developing in the bone marrow, then other organelles are present like nucleus and mitochondria, endoplasmic reticulum. But these organelles in the later stages of development are extruded from red blood cells, and hence red blood cells has more and more hemoglobin packed within it, so that its oxygen carrying capacity is increased. So just to summarize some important features which increase the oxygen carrying capacity of red blood cells and also the oxygen diffusion. Number one, these are biconcave discs, which means that hemoglobin will be very close to the surface. So oxygen and carbon dioxide, they can easily diffuse as compared to spherical cells. Number two, these cells, they have a lot of excess membrane because of their biconcave shape. As compared to spherical cells, these cells can easily squeeze when passing through narrow tortuous capillaries, and hence they do not rupture. So these biconcave cells have less fragility as compared to spherical cells. Not having nucleus and other organelles also provide more room for hemoglobin to be packed inside red blood cells. All these factors increase the oxygen carrying capacity of hemoglobin by packing more hemoglobin inside it, and also the diffusion of gases is increased. Another very important parameter of a full blood count or complete blood count is that you should know the concentration of red blood cells, which in males is slightly higher as compared to in females. It's 5.2 plus minus zero point 0.3 million per millimeter cube in males. In females, it's 4.7 plus minus zero point 0.3 million per millimeter cube. On average, you can say that this is about 5 million per millimeter cube. This millimeter cube unit can also be called as a microliter. So please don't be confused. So you can see here how concentrated our blood is with red blood cells. This is why when we are calculating red blood cells under a microscope using Newport chamber, we dilute these red blood cells first up to 200 times and then we count them. And after counting, then we multiply it again with 200 dilution factor to get the actual count. This is such a big count of red blood cells in our blood that 1 ml of blood contains about 1 billion red blood cells. This count of red blood cells is further increased at higher altitudes. So those living at higher altitude, they are exposed to relatively low oxygen, which increases the production of erythropoietin hormone, and that stimulates our bone marrow to increase the production of red blood cells. So you can call this condition as a physiological polycythemia. A very interesting fact, if someone asks you what is the ratio between the count of red blood cells and white blood cells, what is your answer? The answer is 700 to 1, which means red blood cells are 700 times more numerous in our blood as compared to white blood cells. White blood cells, their count is like 4,000 to 11,000 per millimeter cube. Another important fact, you already know that hematocrit in males is about 45% on average, in females it's 42%. Let's suppose if you are having a normal hematocrit content, what is the total hemoglobin content in your packed red blood cells? The answer is 34 grams per deciliter. I'm not talking about whole blood, just the packed cells. So packed cells, they have about 34 grams of hemoglobin per deciliter, and this is the maximum limit up to which the hemoglobin can be packed inside red blood cells. Now what about the hemoglobin content in your whole blood? This is another parameter of the full blood count or complete blood count. You can see here in males, its value is between 13.5 to 17.5 grams per deciliter. In females, the Hb content is 12 to 15 grams per deciliter. So females have slightly less hemoglobin and also slightly less number of red blood cells as compared to males. So males have more hemoglobin content and a higher number of red blood cells. Why it is so? It's because the sex hormones in males are different than the sex hormones in females. It's testosterone in males and it's estrogen in females. Testosterone is an anabolic hormone which builds muscles. Because males, they have more muscular content and more proteins in their body, these muscles, they need more oxygen for their work. And in order to provide more oxygen, male need more red blood cells because there will be production of more erythropoietin hormone from kidneys and liver, and this erythropoietin stimulates bone marrow, and hence bone marrow will produce more red blood cells. In children, the hemoglobin content is between 11 to 16 grams per deciliter, and in pregnant women it is 11 to 14 grams per deciliter. So it means a pregnant female, when will have a hemoglobin level less than 11, will be called as anemic, and a non-pregnant female, when will have a hemoglobin content less than 12 cutoff value, will be labeled as anemic. A very interesting feature to note that how do we calculate the oxygen carrying capacity of blood? Each gram of hemoglobin carries 1.34 ml of oxygen. So if you have 15 grams of hemoglobin per deciliter of blood, multiplied by 1.34 will give you an answer of about 20 ml of oxygen carried by 1 deciliter of blood. It means if your hematocrit is normal, your hemoglobin content is normal, and if your hemoglobin content is about 15 grams per deciliter, then multiplied by 1.34, which is the oxygen carried by 1 gram of hemoglobin per deciliter of blood, then the total oxygen which your 100 ml of blood can carry is 20 ml, which means 1 by 5. So 1 volume of oxygen can be carried by 5 volumes of blood, 20 by 100, which means 20 ml of oxygen will be carried by 100 ml of blood. This is called as your oxygen carrying capacity of blood. And this oxygen carrying capacity of blood will be increased in case of polycythemia and decreased in case of anemia. Now let's talk about the hemoglobin protein, which is carrying oxygen within red blood cells. This is a conjugated protein which is naturally colored. Hemoglobin protein is made by a protein part which is called as globin and non-protein part which is called as heme. There are 4 polypeptide chains present in hemoglobin. These are 2 alpha chains and 2 beta chains. Each polypeptide chain is having a non-protein group which is called as heme group. This heme group in the center is having iron. Iron is the element which attaches with the oxygen with a weaker bond reversibly, and this bond is called as coordination bond. Each heme can bind with one molecule of oxygen, and because there are 4 heme molecules having 4 iron atoms, so one hemoglobin when fully saturated can bind with 4 oxygen molecules. Because one red blood cell is having a maximum of 250 million hemoglobin molecules, so it can bind with 250 multiplied by 4 up to 1000 millions or 1 billion molecules of oxygen. Very interesting to note that one red blood cell can carry up to 1 billion oxygen molecules when it is fully saturated. So hemoglobin molecule is like a car which is having 4 seats. When all of these 4 seats of hemoglobin they are occupied with oxygen, then we call it oxyhemoglobin. It is having a scarlet red color, and this oxyhemoglobin is present in our arterial blood, which is coming from lungs after being fully oxygenated. When this hemoglobin passes through our tissues, then one oxygen may be given to the tissues. Then this is partially deoxygenated hemoglobin, which is having a bluish or purple red color. So that's why venous blood is having purple red color. Now let's talk about various types of hemoglobin. Depending upon the arrangement of amino acids and the number of amino acids, there are different types of chains of hemoglobin. Although each hemoglobin molecule contains 4 chains, but the types of chains in each hemoglobin molecule can vary in the intrauterine life and extrauterine life. In embryo, there are 2 types of hemoglobin, Grover 1 and Grover 2 hemoglobin. Grover 1 is having 2 zeta and 2 epsilon chains. In Grover 2 hemoglobin, there are 2 alpha and 2 epsilon chains. In the fetus, predominant hemoglobin is hemoglobin F, which is having 2 alpha and 2 gamma chains. After the baby is born and also during adult life, the most abundant hemoglobin present in our blood is hemoglobin A, which is having 2 alpha and 2 beta chains. Its proportion is more than 95% in our total blood. In adult persons, there is another type of hemoglobin which is called as hemoglobin A2. Although its production starts in the late intrauterine life, but also in children and adult persons this is present, and its proportion is 1.5 to 3.5%. It's important to note the percentages and types of these hemoglobins in different lives like intrauterine and extrauterine life, because these chains of hemoglobin and also their percentage can vary in different types of thalassemias. I will also mention 2 abnormal types of hemoglobins. One is hemoglobin S, which is seen in sickle cell anemia. In this case, the hemoglobin is alpha 2 beta 2 S, which means in this case the beta chain of hemoglobin is defective. At position number 6 of the beta chain, glutamic acid, which is normal acidic amino acid, is replaced with valine, which is non-polar amino acid. In case of hemoglobin C, the hemoglobin chains are alpha 2 beta 2 C. Once again here the beta chain is abnormal. At position number 6, glutamic acid, which is acidic amino acid, is replaced with lysine, which is a basic amino acid. So even this small minor change at one amino acid will cause this hemoglobin to become dysfunctional and its features will change. We know that oxygen is the most important molecule that combines with hemoglobin to be transported. Now let's talk about what other molecules can bind with hemoglobin. One of them is carbon dioxide. 23% of the total carbon dioxide which is transported through blood binds with hemoglobin to be transported from the tissues to our lungs so that it's expired out. You know that hydrogen ions are produced in our tissues from carbon dioxide because carbon dioxide is an acid which is produced in response to metabolism. This carbon dioxide it generates hydrogen ion and this hydrogen ion must be buffered. Much of the buffering capacity is provided by plasma proteins because they are negatively charged at our normal pH 7.4. So just like our plasma proteins, hemoglobin also provides the buffering capacity. But because the concentration of hemoglobin is much more than the concentration of plasma proteins, hemoglobin provides more buffering capacity as compared to that of plasma proteins. Almost 50 to 60% of the total buffering capacity of blood is provided by our hemoglobin. Hemoglobin can also bind with carbon monoxide. Normally, carbon monoxide is not present in our blood. But in some cases, when carbon monoxide is there in our environment, this can enter into our body and can bind with hemoglobin with a very high affinity. Please note that carbon monoxide binds with hemoglobin with an affinity which is 250 times more than affinity of oxygen to bind with hemoglobin. And in this way, the carbon monoxide leads to poisoning. Another important molecule which can bind with hemoglobin is nitric oxide. Nitric oxide binds with the hemoglobin in our lungs and then delivered to tissues. Nitric oxide is a very important vasodilator substance. When nitric oxide is given to the tissues, there it causes dilation of our arterioles and then more oxygen will be given to our tissues. So just to summarize, other than oxygen, what can bind with hemoglobin and can be transported in our blood? Carbon dioxide, hydrogen ions, carbon monoxide and nitric oxide. Now let's talk about some important enzymes which are present inside red blood cells. Two important crucial non-renewable enzymes are present inside red blood cells. One of them is the glycolytic enzymes and the other one is carbonic anhydrase enzyme. In order to produce ATP, red blood cells cannot use their oxygen. It's very funny because red blood cells they are filled with a lot of oxygen but they cannot use that oxygen. Why it is so? It's because red blood cells they do not have mitochondria. And why these red blood cells should not have mitochondria? So that more room is provided for packing more and more hemoglobin inside them. So when red blood cells cannot use the oxygen which they carry, how will they produce the ATP? It will be by anaerobic glycolysis. And why red blood cells need ATP? It's because they need ATP for sodium potassium ATPase pump, and this pump is very important, number one, for maintaining the concentration gradient for sodium and potassium and also for maintaining cell volume. If ATP is not available, this pump cannot work. This cell will become bigger and bigger in size and it will burst. The second enzyme which is present inside red blood cells is carbonic anhydrase enzyme. This enzyme is important for this reaction to take place in our tissues. Carbon dioxide, which is produced in response to metabolism in our tissues, binds with water to make carbonic acid. Carbonic acid is an unstable compound which dissociates to make hydrogen and bicarbonate ion. Hydrogen, which is an acid, it will be buffered by hemoglobin, and this bicarbonate, it is the primary form in which carbon dioxide is transported in our blood plasma. Bicarbonate will come out of red blood cells in exchange with chloride, and this exchange is called as bicarbonate chloride shift or hamburger phenomena. This bicarbonate in this way it will be transported in our plasma to our lungs where the reverse process takes place. Then this is converted again back to carbon dioxide and carbon dioxide will be expired out. So just to summarize, we can say that red blood cells transport carbon dioxide in two forms. One is hemoglobin binds with carbon dioxide and this transports about 23% of the total carbon dioxide. About 70% of the total carbon dioxide is transported in the form of bicarbonate ions within our blood plasma. The rest of 7% carbon dioxide is actually dissolved within our blood plasma. So just to summarize, in today's session we have studied some important parameters of full blood count. You should know their values in males and females and also the units. Some students make mistakes when they are presenting these values. They forget the units or they give the wrong units. We have also discussed the structural features of red blood cells and how they increase the oxygen carrying capacity of blood. We have also discussed the chemistry of hemoglobin and also various types of hemoglobin in healthy and diseased persons. And then we have discussed what are various types of molecules other than oxygen which can be transported by hemoglobin. And then in the end, we have discussed two important types of enzymes which are present inside red blood cells. And these are glycolytic enzymes and carbonic anhydrase enzyme. If someone asks you what are three important functions of hemoglobin, you should say number one, it transports oxygen by binding with hemoglobin. You should also say hemoglobin also transports carbon dioxide. And third important function of hemoglobin is to bind with hydrogen and buffer it and to prevent change in our blood pH. I hope you must have enjoyed this session. In our next session, we will discuss the erythropoiesis or production of red blood cells, and then what are the factors like erythropoietin hormone which control the production of red blood cells. Thank you so much for watching this video. See you next time with another video.